All right, the chemistry of microbiology. So chemistry is the study of matter and all things matter. Matter is anything that has mass and occupies space. So everything is considered matter except for energy. Elements are the most basic building blocks of matter, and an atom is the smallest possible piece of an element that still retains properties of that element. Atoms are made of things like electrons, which are little negatively charged particles around the outside of the nucleus of an atom. And then inside the nucleus, you have protons, which are positively charged, and neutrons, which are neutral. You guys have all seen a periodic table before. All of the elements that we know exist are in this periodic table. It tells you the names of all of the elements, their symbols, and a little bit about them. When we're talking about microbiology, we have some elements that are essential for life and some that are not necessarily essential, but important, all right? The heaviest hitters are carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, collectively called SNOPs. The others are our trace elements. These are things that are needed by cells, but only in small amounts. Sodium, potassium, magnesium are electrolytes, zinc and iron, calcium, copper, molybdenum, cobalt, manganese, vanadium, all of those are in one way or another important for our cells. Carbon is kind of the molecule of life. We are carbon-based elements. What does that mean? Well, that means that carbon is what primarily makes up our cells and our macromolecules inside our cells. Why carbon? Why carbon? Why does it have to be carbon? Carbon has in its electron shells, which are these little orbitals of electrons that exist outside the atom, it has 4 electrons. Okay, 1, 2, 3, 4. Now, atoms want to have 8 electrons in their outer shell to be happy and stable. So carbon has half of them. So it will share up to 4 electrons with other atoms in chemical bonds to get to that number of 8 electrons. And where am I going with this? Well, carbon is very small, so its size allows it to be bonded to many different things and many different molecules. And the fact that it can pair up with 4 different partners at the same time means that it can make some very complex structures. Okay, so carbon is small and versatile, and it likes to share electrons, so it likes to make chemical bonds in these big molecules, and that makes it really conducive to life and living things. So we say that any kind of chemical or molecule that contains carbon and hydrogen is what we call an organic molecule, or a molecule of life. If it is a molecule without carbon, they're called inorganic. Okay? Carbon can bond, like I said, 4 things, and they can even bond with other carbon molecules and make these big rings and chains and all of these really complex structures that you really don't get with any element except carbon. Our inorganic molecules are important too, though. They are made of, they're things like salts, water, small molecules. They don't necessarily form the structure of the cells like carbon compounds do, but they're all very important. In our cells, we have sugars or carbohydrates, we have lipids, amino acids, DNA, all of those have carbon in them. Okay? They're very large and complex structures, and they make up our cells. So that's why carbon is our organic molecule. Okay, there's a video here that I'm going to put in the module on Canvas. This is about carbon and why it's so important. And let's talk a little bit about the chemical reactions that put the molecules together in our cells, and then we can actually talk about the macromolecules themselves. So chemical reactions involve making and breaking chemical bonds between atoms. There is always an energy transfer that occurs during chemical reactions. When a chemical reaction happens, the participants in that reaction will either absorb or release energy, right? You may have heard of endergonic and exergonic before. If it's endergonic, it's going to absorb energy. If it's exergonic, it will release energy. And that'll become more important when we talk about metabolism. There are two kinds of reactions that happen in our cells that are super important because they're pretty much the reactions that make and break things in our cells. A synthesis reaction is when you take things and put them together, right? You're synthesizing something, you're combining them together. In our cells, we call that an anabolic reaction. And that happens through a process called dehydration synthesis that we'll talk about more later. The opposite of a synthesis reaction is a decomposition reaction when you're breaking things apart, right? In our cells, we call that catabolism. And that is going to be really important in cell metabolism and respiration. And that reaction is by hydrolysis, which we'll talk about later. But just to introduce those terms now. A little bit about acids and bases and pH because it's important in medicine, even though your book doesn't really go into it. When you have a solution that's a liquid, you either have an acidic or basic or neutral solution, right? So if your solution is acidic, it's going to have lots of little hydrogen ions floating around in it. If it's basic, you're going to have lots of little hydroxide or OH ions floating around in it. Okay? And then salts are essentially solutions of things that are not H or OH. So when we're talking about pH, it's actually a number that indicates how acidic or basic a solution is. Right? A pH 7 is neutral. If it's less than 7, it's acidic. If it's more than 7, it's basic. Most organisms are going to grow in the neutral to slightly acidic range, 5 or 6.5 to 8.5, somewhere in that, the range close to neutral. This is important because our enzymes and other molecules require very specific pHs to have their shape and their function. In medicine, a lot of times we'll use buffers, and in cell culture as well. Buffers are special solutions that help prevent changes in pH that can happen. All right, but what we're really going to focus on are the molecules of life and what they're made of. So carbon is kind of the most basic thing, right? Carbon can do lots of different things. It can bond in lots of different ways. When you have a lot of carbon together, a lot of times there will only be very subtle differences between molecules. And those subtle differences will be what we call these functional groups. They're specific groups of atoms that give chemicals different properties. And they're found attached to usually these big carbon molecules. For example, an OH group is an alcohol group. So things like methanol, ethanol, isopropanol, they're really just these big carbon and hydrogen compounds with an OH on them, making them an alcohol. Your book goes into functional groups a lot. We're really not going to focus on them. Here's an example of some of the functional groups. One of the molecules that we're going to pay attention to, though, are amino acids. So amino acids make up our proteins. They have a couple of different groups on them. They have an amino group, your carbon in the middle, a carboxyl group on the end, and then on the other side of this carbon group, they're going to have an R group. And each of the 20 amino acids has a different R group. The entire rest of their structure is the same, though. Okay. All right. So what our focus is going to be are macromolecules, which are polymers made of little units that are repeated over and over again. Those little units are called monomers. So we have carbohydrates that are made up of monosaccharides. We have lipids that are made of fatty acids and glycerol. We have proteins that are made of amino acids. And we have nucleic acids that are made of nucleotides, like in our DNA. Okay? Carbohydrates, lipids, proteins, and nucleic acids are our 4 macromolecules in biology. Okay. Here is a good summary table of their functions and the macromolecules. And you already know probably a little bit about them all. When monomers are joined together to make that big macromolecule polymer, they're joined by dehydration synthesis reactions. What that means is that one molecule has a bunch of atoms and it ends with a hydrogen. And then another molecule has a bunch of atoms and then it ends with an OH group. In a dehydration synthesis reaction, the hydrogen comes off from one molecule and the OH comes off from another molecule. When you combine them together, you get 2 hydrogens and an oxygen, or H2O, or water, hence the term dehydration. You're removing a water molecule. And in their place, you have these 2 kind of open, sticky ends of the molecule that stick together and make one big molecule. That's called a dehydration synthesis reaction. That's how our cells build our monomers into our polymers and our macromolecules. And the opposite of that is hydrolysis. You start with a water molecule, you break that into a hydrogen and an OH, and you use that to break your big molecule into 2 smaller ones. All right, let's talk about carbohydrates. Carbohydrates are very important for cell structure and for energy sources. They are made of carbon, hydrogen, and oxygen. Okay, we've got things like monosaccharides. Those are our simplest sugars. They have usually between 3 and 7 carbons, things like glucose, fructose, and galactose. Sometimes we have 2 sugars put together in a molecule called a disaccharide. Disaccharides themselves can be monomers for different polymers as well. Sucrose, lactose, maltose, those are all disaccharides. Is what they look like in the bigger picture. Okay. So we have our simple sugars like our monosaccharides and our disaccharides, and then we have our more complex carbohydrates, which are made of lots of either monosaccharides or disaccharides put together into really big molecules. So complex carbohydrates or polysaccharides are our big polymers of sugar. Interesting thing about our complex carbs or our polysaccharides, they're not sweet. They don't taste sweet like the simple sugars do. They're also not soluble in water. Think about cellulose. Right? Cellulose is in the cell walls of plants. It is a very tough, strong structure. It's hard to chew. It's hard to digest. It's not sweet. Okay. Things like starch. Think of potatoes. Right? Potatoes are not sweet. Starch is essentially a way that plants can store carbohydrates, but they're all made of essentially glucose into different kinds of polymers. Chitin, that really strong substance in fungal cell walls and insect exoskeletons, that is a polymer of 2 repeating sugars. The next macromolecule are our lipids. Lipids are for energy storage. They're very important in our cell membranes. They are not soluble in water. Lipids are made of fats or triglycerides. Again, carbon, hydrogen, and oxygen. A lipid contains 1 glycerol. This is the glycerol molecule up here. And between 1 and 3 fatty acids. Fatty acids are these long chains of carbon with hydrogens all around them. Again, they're formed by dehydration synthesis. With our lipids, we can have saturated fats and unsaturated fats. And here's something that's really interesting and relevant to our everyday lives. We all know what saturated fats are when we talk about our diet, right? They're bad for us. They're saturated because all of these carbons have hydrogens bound all around them. Okay, so they're all completely full. Okay, there's no carbons bound to other carbons. Every carbon is completely maxed out with hydrogens. It's saturated with hydrogens. Saturated fats are things that are usually solid at room temperature, like butter, right? Unsaturated fats, you don't have hydrogens surrounding every carbon. When a carbon is not surrounded by hydrogens in a fatty acid tail, it will make two bonds with the carbon next to it instead of binding to a hydrogen, and it creates kind of a kink in the chain, making a more fluid molecule. These guys are usually liquid at room temperature, things like olive oil. Then we have our really complex lipids. They're, again, carbon, hydrogen, and oxygen, but they have either phosphorus or nitrogen or sulfur also attached to them. Our membranes are made up of phospholipids. This is probably the most relevant complex lipid for all of us. Phospholipid is our lipid, which is our glycerol with two fatty acid chains and a big phosphate group on the top. So it's a phospholipid, meaning it's phosphorus and lipid together. It makes up our cell membranes because this phosphate group likes water and the lipid doesn't, right? So you have the polar head and the nonpolar tails, polar meaning it likes water and nonpolar meaning it doesn't. In our plasma membranes, when you have a lot of these phospholipids together, they arrange themselves into this bilayer where all of the fatty acid tails are on the inside and they seal off the water from coming in and form a really good barrier. And on the outside, you have all of the phosphorus groups. So the phosphorus groups will interact with the water and the fatty acid tails don't. Steroids are also lipids. They're also referred to as sterols. They're kind of 4 carbon circles put together, and they're also in our membranes, in eukaryotic membranes anyway. Our next macromolecules are proteins. They are really, really important for cell structure and function. All of the macromolecules are important. Proteins are really, really important. Proteins make up all of our enzymes, transport proteins, cell structure proteins, things that help us with movement. Some bacterial toxins are proteins, hormones are proteins, antibodies made by our immune system to help fight off pathogens, those are proteins. Proteins are very, very important. Again, made of carbon, hydrogen, and oxygen, and nitrogen, and occasionally sulfur. Proteins are made of amino acids, right? You guys probably already know that. There are 20 different amino acids. Here's their structure. Again, you have an amino group on one side, a carboxyl group on the other side, one carbon in the middle of all that. One of the bonds that carbon makes is to a hydrogen, and the other one is to this R side group. And that R side group can be, is different for each of the different 20 amino acids. And that's what gives the amino acid its specific structure and function. Proteins are bound together by what we call peptide bonds, which are just fancy dehydration synthesis reactions. Here are the 20 amino acids. You don't need to know this, but just so that you can see, they're all the same except for that R group, right? There are different levels of protein structure. You have your primary protein structure, which is just the sequence of amino acids that you have. Once you have your primary structure made, it starts folding up or curling up into helices or pleated sheets. That's your secondary protein structure. Those fold up into these kind of big 3-dimensional globs. That's your tertiary structure. And sometimes you have multiple tertiary structures that associate together, and that's called quaternary structure. But a protein structure is super important because its structure is specific to its function. Without its structure, it can't perform whatever function it's supposed to have in our cells. So while it might look like a giant glob of amino acids, it's actually very highly ordered and specific. Same picture, different slide. Sometimes you have proteins that are bound to other things, other macromolecules like glycoproteins, nucleoproteins, lipoproteins, alloproteins. They can make combinations with other molecules. When a protein loses its really specific three-dimensional structure, we call that denatured. Okay, think of it turning into like a wet noodle. Okay, sometimes that denaturing is reversible. Sometimes it can fold back up into its really specific structure, and sometimes it can't. Things like high temperatures can denature a protein. Really extreme acidic or basic pHs can denature a protein. The amount of salt present in solution can denature a protein. So proteins are very specific. They're also pretty sensitive to their environments. The final macromolecule are nucleic acids, RNA and DNA. You guys are familiar with them already. They store genetic information. Our nucleic acids are made of nucleotides. Each nucleotide has 3 parts: a phosphate group, a pentose sugar, which is a sugar made of 5 carbons, and a nitrogen-containing base. In the pentose, if you have DNA, it's deoxyribose, that's the name of the sugar. If you have RNA, it's called ribose. That's kind of the big difference between DNA and RNA. And then you have your nitrogen-containing bases, A, T, C, or G. A is adenine, G is guanine, T is thymine, and C is cytosine. If you have RNA, instead of T, you have U for uracil. Okay, those are really the only differences. So DNA makes pairs from one nucleotide to another. A will pair with T and C will pair with G. And it looks like that sugar and phosphate group make up this kind of backbone string. It's called the sugar phosphate backbone along the DNA. And then the nucleotides kind of stick out from that. And they stick into the DNA structure, and that's where they can bond with one another. Between these nucleotides, we have hydrogen bonds. Okay, A binds with T, C binds with G, because A and T can make 2 hydrogen bonds, and C and G can make 3. So 2 bonds with 2 and 3 bonds with 3. The strands are always complementary to each other in that one strand will have a sequence and the other strand will have the exact opposite of that sequence. The orders of the nucleotides tell us what amino acids are going to be made from them, and they determine all of our traits. That's what makes up our genes. We'll talk more about them when we talk about genetics. Here's a picture of RNA, just like DNA, except the sugar is different. And occasionally, instead of a T, you'll have a U. There's 3 kinds of RNA. They all have very specific functions, pretty much all related to making proteins. You have messenger RNA, ribosomal RNA, and transfer RNA, and we'll talk about this later. Also in the nucleotide category, and super important, is ATP. ATP is our energy in our cells. It's our energy source. It's called adenosine triphosphate. It is in the nucleotide category because it looks just like a nucleotide. It is a nitrogenous base, adenine, the A, only that one, bound to a ribose sugar, bound to 3 phosphates if it's ATP. When it releases that energy, it becomes ADP with 2 phosphates. Okay, here is a great summary of our monomers and polymers. We have carbohydrates made of monosaccharides, lipids made of glycerols and fatty acids, proteins made of amino acids, and nucleic acids made of nucleotides. There's a couple other really great videos here for you to watch about the macromolecules, and that is the end of our lecture.